Hello everyone, the article I will be presenting is mRNA targeting to endoplasmic reticulum precedes ogoprotein interaction and microRNA mediated translation repression in mammalian cells. So this is written by two names that I cannot pronounce. It's from the Journal of Biological Chemistry. It has an impact factor of 4.106 and it was published in 2015. Here is just a quick overview of the steps I'll be going through in this presentation. I'll start with the purpose of their article, the background, some terminology, methods, results, and I'll finish up with conclusions. So we know from lecture that mRNA passes through various steps on the rough endoplasmic reticulum to get synthesized into protein. However, what we didn't learn is that sometimes there's this repression step that occurs, and this is mediated by miRNA. So they knew going into this study that miRNA binds to the mRNA and represses protein synthesis. However, the purpose of this study was to actually delineate how a target mRNA passes through the RER before miRNA mediated repression actually sets in. So they wanted to figure out the actual sequence of events and also localize where this was occurring around the RER. So throughout this, try to think about how it ties into what we already learned in class and builds on that. So that you can follow along a bit better, I'm going to go ahead and tell you the results, so that way hopefully it's easier for you. Knowing this in the beginning, you can follow along better understand how this breaks down. So the result of this was that the first event is mRNA localizes to ER-bound polyribosomes, and then this protein and this protein complex that I'll go into later, called OGO2 and miRNPs, they bind, and then there's translation repression. So this is what they determined through their research. This is also compartmentalized in the RER to optimize the miRNA targeted message and the complex components on the ER. So now I'll just do a little background so you can understand a little bit better. So I'll talk about what actually microRNAs are. So microRNAs start in the nucleus in a hairpin form called pre-miRNA, which you can see at the top of the figure. This then gets exported out of the nucleus, and then an enzyme trims the pre-miRNA and removes the hairpin loop. This leaves a double-stranded miRNA, and one of these strands leaves and forms an miRNA complex with ogoproteins. So you can kind of see that on the left. You can see the blue strand highlighted, and then it leaves and it forms this complex with the red. And RISC is just another name for this complex that it forms with. So the complex, along with OGO2, it also involves miRNP, and so this together all is labeled RISC. And so then this whole complex bound with the miRNA can actually bind to mRNA. So miRNAs are about 22 nucleotides long, and they base pair, as you can see in the figure, with the 3' UTR region of mRNA. And this binding region is a target sequence, so they're very specifically looking for this sequence. And the binding in this location allows for gene regulation. And the binding is not completely complementary, which you can see in the figure with the little loop in the middle of the blue strand. That's where it doesn't exactly match up, so it's not completely complementary, but it's enough to bind. And the binding of this blocks ribosomes, and it blocks the continuation of translation. So it stops that translation of the mRNA into protein. So we know from class lecture that some mRNAs are translated through ribosomes bound on the RER. And we know, or there have been many studies done, to confirm this. And through other studies, they have found a lot about mRNA. There's information on the organelles that control the trafficking of it. There's information on how it co-sediments with polyribosomes and other molecules. But there's not much research before this one about how it localizes and the steps that occur. So that's what this one kind of focuses on. Okay, so now I'll start going through some terminology so that way throughout the rest of the presentation you're not completely lost. So miRNPs instead of RNAs are this huge, this big complex that binds to the miRNAs. This is also known as RISC, as I showed in the previous slide. And this is basically a bunch of proteins that are associated to miRNA, along with the AGO. So AGO and some other proteins form together to form this whole RISC complex, which you can kind of see in the figure. AGO starts on this, but then once it actually binds to the mRNA, then it is known as this RISC complex. So miRNPs, and for the rest of this presentation, just basically is referring to this big complex of proteins that work to allow miRNA to do what it's going to do. Ogoproteins are these highly specialized small RNA proteins, or binding proteins, and they act together with the protein binding partners to interfere with the translation. So they're what's actually doing the interfering with the target sequence of mRNA. LET7A is, I'll refer to this later in the presentation, it's actually the gene that encodes mRNA, so throughout their results and such, they don't say miRNA for a lot of it, they just use this gene, this LET7A gene, and that's indicative of miRNA. And then thapsigargin, maybe, is the inducer of ER stress, so it inhibits an ATPase of the ER that affects calcium homeostasis. So this just basically means that the endoplasmic reticulum is in stress. So just a little summary about those first two. Proteins join with the ogoproteins to form the RISC complex / miRNP, same thing. The whole complex then guides the miRNA to the target sequence. And the miRNA is what actually binds, incomplete complementarily with the target strand, and the ogoprotein is what is actually interfering with the translation. Okay, so I'm not going to go into the details of the experimentals, like the methods, because it's really if you look at the article, it's really just the nitty-gritty. They have little sections titled all of these, and they really just go into the very specific details, like this is how much reagent we used, this is how long, kind of things like that, so I don't want to bore you guys. But some of them you can recognize right off the bat, like PCR from other classes, and some of the techniques I will point out while I go through the results of this experiment. But here's the kind of the summary of the different methods that they used. All right, moving on to the results. So their first results center around localizing the compartments where all these different molecules, like the miRNAs, ogoproteins, and the target messages are. So they really wanted to first start off by localizing it. So they started, you can kind of see on the left, they started with HEK 293 cells, which are human cells, and they treated them with detergent. This permeabilized the plasma membrane selectively, so where there was high cholesterol is where they formed the pores. and this resulted in a soluble and insoluble fraction. The insoluble fraction had an ER marker protein, so they knew that ER was there in this insoluble fraction. So within that fraction, those fractions, if you just follow along on the images going down the row, there was the AGO2 protein found. And so just remember that this is part of the miRNA complex that is doing the repression work. And so then they just confirmed this, that the AGO protein was within that fraction. So they confirmed this with and B on there. I won't bore you with all the details, but that was just them confirming that AGO2 was in the RER. And then they wanted to confirm association of miRNA and AGO2 protein with the RER. So they isolated more of these cells and observed most of the AGO2 and miRNA, which is indicated by LET7A, like I talked about before. So if you look at like image C, it says LET7A, so that means miRNA. And then they also looked at AGO2 and some other proteins. So then this is just confirming. You can see like on the right side of that image in C that the AGO2 and the LET7A are both a lot darker on the RER side, and the polysome. So this was just confirming their association, and then then the rest of these are basically the same type of idea. They're just continuing the verification of the association of AGO2 and miRNA with the RER. They also confirmed that the association of these to the ER is based on the ER function. So the TG that I went over in the terminology is known to affect protein translation. So they showed that when TG was applied, there was a reduction in the miRNA association. So you can see that in K. So when there's no TG, then it's pretty much the same. But when there's a lot of TG in the RER, the mRNA association is much lower. So the function of the mRNA and the mRNA complex is really dependent on how well the ER is held up. So when that TG is applied, which remember it causes ER stress, then it's a lot harder for the mRNA to associate. So just a reminder, we are interested in how this occurs sequentially. So like understanding the association allows them to kind of localize their studies and better like have a starting point for figuring out the sequentials of this. So that's kind of what this is doing. It's kind of localizing and then setting them up for the rest of the experiment. So then they wanted to characterize the RER associated miRNPs and target messages. So they isolated the polysomes associated with RER and found most of the miRNA, AGO2, and target messages in ribosome compartments in the supernatant. So this confirmed that you can see in like part A, the supernatant is the ribosomal pool, so they wanted to confirm they wanted to confirm that all these components of the miRNP complex are actually associated with the polysomes rather than directly with the ER. So then they continued this with other techniques as well, which is kind of being shown. And these showed that all this mRNP complex components are localized to the polysomes that are associated with the RER rather than the ER itself. So again, they were interested in the localization and steps of the repression of the synthesis. So this further determines the location and helps set up for better studies for the figuring out the actual sequence. So then their next objective was, of course, to actually determine the sequence of the ER targeting and mRNA repression. So they made an expression system where they had a reporter mRNA that was dependent on a drug. And so when they added the drug, the mRNA level increased sharply in total and in the RER. And this is kind of shown in some of these figures. They wanted to see if agoproteins were bound before or after the mRNA was targeted to the RER polysomes. So they measured the targeting of these things in the presence and absence of mRNA, and they found that translation repression occurred 8 hours, at least 8 hours after when mRNA has bound to the complex. So this confirms that the binding of this AGO2 and mRNA definitely comes before the miRNA repression of the mRNA. So basically, all this showed that all these components definitely all get enriched in the ER membrane. So the mRNA targets, the miRNA, the AGO2, the whole complex, they all get enriched in the ER membrane. So this is the site where the mRNA gets associated with the miRNA and the AGO2. They also found that the sequence of events is that the mRNA is destined to the polysome first. So you can see this in this image that target mRNA is destined to go towards the ER-bound polysomes. Then the AGO2 miRNA RNP complex that targets that mRNA will associate. So you can see that following along the image, it actually associates to that target strand. And then finally, the repression occurs. So this is the very last step. So after it binds, then the repression actually begins. So ultimately, as the miRNA complex is part of gene regulation as it represses, understanding the sequence of these events and localizing it can help researchers better understand gene regulation. And this study itself did not offer up any advantages to this knowledge or possible future experiments. So I thought this could be a good thing for you all to speculate on in the comments, like what could be some advantages to understanding how this gene regulation and repression is occurring and the sequences of it. What could be some advantages of that? And then so lastly, I didn't really notice any weaknesses in the article. It was pretty easy read. I think maybe they could have done a bit better with their methods because, like I said earlier, they were just really dense and very precise details without any explanation of what they were tying it into. So maybe they could have done a little bit better with that. But maybe that's just a characteristic of science articles. And then another maybe weakness is that there were only 2 articles. I notice a lot of these have a ton of articles, or a ton of authors, and so maybe that could have been a weakness, more authors, more perspectives and more ideas, but at the same time, maybe that was a strength of theirs. They only had 2 people, no fluster. But thank you, that's all I have for you.